Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome, everybody, to the Genetics Podcast. I'm really excited to be here today with Dr. Samuli Ripatti, who is the director of the Institute for Molecular Medicine Finland, also known as FIMM, to those listeners who are voracious consumers of genetic biobank data. He's also professor of biometry at the University of Helsinki and has affiliation with the Broad Institute in Boston as well, where he's led some major cross-country collaborations, international collaborations, looking at both common disease risk through polygenic risk scores and other modern techniques, but also looking at rare and ultra rare variants, especially in Finland, where you have this amazing population, actually, where you have great historical medical records, but also a population structure that makes studying some particular diseases really impactful. So thank you so much for taking the time to join us today.
Thanks for having me. Great to be here.
I would love if you could take us back to your origin story in genetics. What got you interested in the first place? And is there a common thread that brings us through to today, or is it a winding path the whole way?
So my background is really in statistical methodology, and that's what I did my PhD for, mathematical statistics in Stockholm University. And during that period, I really got more involved with first quantitative genetics, the twin studies, and the way twin registries in the Nordics and elsewhere can be used to understand at least some corner of genetics. And then after that, I got first. I was a couple of years outside of academia after my PhD, working with internet technologies and which were hot in the early 2000s. And then I thought that maybe it's time to get back to academia. And I got involved with Leena Peltonen, a Finnish famous geneticist, and did a postdoc in her group in Helsinki. And that's how it all then started. This was right at the time when genome-wide association studies started. So we had a tremendously large the lipid genetic study with 1,500 twins from all over Europe at the time, and that's how it all got started.
I have to ask about Leena Peltonen, because I, back in my PhD days, went to the Leena Peltonen School of Human Genetics, which was an amazing couple of days. That if people aren't familiar with it, it's really unique. It brings together some high-powered academics and industry individuals, like I'm sure you've been a tutor there yourself in the past, as well as PhD students. And there's an equal number. You get matched with mentors, people who can talk to you about what kind of career interests you might have. And there's a series of lectures basically where everyone who's there presents at least once. And it's a really small group setting. And the people that I met there, I've remained friends with for a long time. And it was set up in her legacy, obviously. What was she like to work with? Because I've obviously never met her personally.
So Leena was really a character in many ways, and a very strong figure in not only in Finnish science but throughout the world. And she was one of the sort of the early adapters to large scale population based studies, and that was one of the areas where she did a lot of work, particularly at the time that I joined the group early on, she worked with Mendelian diseases. And in Finland, you mentioned in your introduction, we have a bit of a bottleneck population with multiple bottlenecks. And therefore, we actually have our own set of rare diseases that are much more common in Finland than in elsewhere else, or some of them don't even exist elsewhere. And these are typically recessive diseases. So Leena was early on mapping those and then moved into common diseases, struggled through the years of candidate gene studies as much of the field, and then we're sort of working on the early genome-wide association studies in 2000 before she passed away. It's great that you mentioned the summer school, because it was actually rebooted this summer. After COVID, it was out for a couple of years, and it just ended a couple of weeks ago. And it is exactly like you said, small group of students, roughly the same number of tutors as students, and a lot of one-on-one discussions and mentoring, and that's the whole idea on top of lecturing and giving talks, for the opportunities to give talks for the students.
Yeah, it's something I'd recommend to anybody who's studying human genetics right now. I'm glad they rebooted it. Take a look and apply. It's some of the best couple days that you could dedicate your time to.
It's probably the best way for a computational genetics or population genetics PhD to find a postdoc place. So that's really one of the keys there.
Yes, that's very good and practical advice. Speaking of transitioning, so you worked in her group, you went out on your own. What were you most interested in? And I'm curious to hear, you mentioned that you went in and out of industry a little bit. I'd love to hear what you started working on, how that carried through to today, and also what you've learned about the power of collaboration, because you have worked on some major multinational programs that have got to scales of more than 1 million individuals, for example, to understand some common diseases and traits in pretty fundamental ways. So what have you learned along the way?
So I started with lipid genetics. That was a good model for trying out large scale mapping exercises. In many ways, it's a good model. One of them was that we already knew at the time when we started genome-wide association studies, we already knew LDL receptors role, so this ApoB was known, and so there were these components that we were expecting to see, and then we started building on that. And obviously at the time when even the big data sets were on current standards pretty small. I mean, these were cohorts with a couple of thousand individuals. It was clear that the only way to really boost the discovery was to collaborate. And it was also a time when the big shot geneticists were divided into two: those who could collaborate and those who could not. Right. And it was the first group of geneticists that then really got into the next phase and were the leaders in the field in terms of the discovery. So I worked a lot first with lipids, obviously with cardiovascular diseases more broadly, with lipids often leading to them. And then it was the time when also the metabolomic screen started to be, or the prices started to come down, so that you could do those in large scale. And that sort of grew into actually hundreds of metabolites, particularly lipids and lipoproteins, but other metabolites as well. And trying to understand the balance between the genetic background of the variability versus then what we eat and exercise and lifestyle factors that are going in there. And this all led then to actually trying to put together the knowledge or discoveries that we've done in order to trying to understand what is your and what is my risk for getting the diseases, and how much can we capture with the discovery. And one of the early on papers that we did in 2010 was putting together 13 SNPs, which was tremendous amount of genetic data. and look in two Swedish and two Finnish cohorts, how well can we predict incident coronary heart disease at the time, and how do these genetic scores, as we call them, or genetic risk scores, do they work together with known risk factors? So in summary, I think overall coronary heart disease and cardiovascular diseases more generally have been a very good model in two ways. One of them is because there's a good amount of knowledge, or was already at the time, both in terms of epidemiological risk factors as well as genetics. So we didn't need to start really from scratch, which is often the case for many other diseases.
Yeah, and this is a good segue, I think, to one of the topics that I want to spend a good chunk of time on today to pick your brain on, because you've done a ton of really interesting work over your whole career, but especially recently, around how do we implement these kind of advanced polygenic risk scores and other risk prediction or stratification tools into the clinic. So, for example, I think earlier this year you had a paper that you published in European Heart Journal on polygenic risk scores in cardiovascular clinics. I'd love if you could just give your perspective of the state of the art today. What do we know about what polygenic risk scores can do for us clinically, and then what are the big questions about what they might be able to do in the future but can't yet do today?
So I think there are three different components that really need to be in place if we want to think about clinical implementation. First of all, we need to be able to capture the genetic risk well enough. So the polygenic risk score needs to be predictive. But that's the case now for many different diseases and many common complex diseases: diabetes, breast cancer, coronary heart disease. Good examples of that as well. The second thing is that what I think makes it much easier for think about clinical implementation is that if there is already a pathway, sort of a pathway in terms of clinical setting, or a time in our lives when we are anyway checked for disease risks, then providing genetic information on top of these routine settings is the way to go, because that's where we already know or have procedures in place. And they're the two examples, I think, where we are furthest is coronary heart disease, in terms of that we have these regular checkups, or we at least should have these regular checkups for the risk, where we measure our cholesterol and blood pressure and ask about our diet and exercise habits, or smoking or alcohol usage, these type of things. And then these have been trained into clinical risk scores in different countries, and providing then additional biomarker or risk factor, if you like, with polygenic risk score is kind of a natural extension. A second example that we have been working with is actually breast cancer, where again we have in many countries there's a mutation screening for BRCA1 mutation, BRCA2 mutation, POP2 mutations. There are panels for these that are applied in certain settings. For example, in Finland, if there's an early onset breast cancer patient coming in with recorded family history, then often the healthy relatives or healthy female relatives of this index case are then screened for these mutations. Now, bringing polygenic risk score on top of the mutation screening is again a natural setting where these can be applied, particularly at least in our hospital in Helsinki, University of Helsinki. Even if we enrich to early onset cases, we search for mutations. It's about 10% that those with family history and early onset cases carry a BRCA mutation or a similar effect mutation. So there's 90% of these cases that do not seem to be at least captured by these known mutations. Now, providing polygenic risk score next to these mutation screening, we can push that up to 30%.
Wow.
With clearly elevated polygenic risk score. on the same level as a sort of a medium level mutation profile. So that's another component.
And is that significantly higher? Sorry to interrupt. In breast cancer
No.
compared to coronary artery disease, for example, do you get that level of explainability from polygenic risk across a broad range of diseases?
I think breast cancer is a little so polygenic risk for breast cancer is a little bit more sort of stronger than coronary heart disease, not much. For coronary heart disease, the baseline is such a good thing. So
Right.
if you take age and sex, if you have a 60-year-old male, the risk is really high for everybody, using our clinical tools. If you have a 40-year-old female, the risk is really low, even if the blood pressure is high, even if the cholesterol is high. But anyway, the current tools are pretty good. So adding on top of that doesn't make such a huge difference than with breast cancer. And that brings us to the third component that I think needs to be there. And this is to do that. Obviously, there has to be something you can do to lower the risk. Without that, it doesn't make any sense to bother people. So for heart disease, it's through medication, through statin use, it's through other or lifestyle changes. We can do that. And again, it's important that both these non-genetic factors as well as genetic factors affect the risk. For breast cancer, it's more difficult to prevent the disease, but the early detection is, of course, really important. And therefore, you can have screening procedures that are personalized to these high risk individuals. They should start earlier, maybe have mammography more often, or other tools of screening. So these are, I think, the three tools that are really the key for applying these in a clinical setting.
Yeah, that's a really good framework. And this last point actually reminded me of one of your really interesting papers that I wanted to ask you about, which was back in 2023, I think early 2023, but you almost ran this process in reverse. The title, for anyone who wants to look it up, is Genetic Predictors of Lifelong Medication Use Patterns in Cardiometabolic Disease. I thought this was a really interesting study design because you looked at you worked backwards from the medication use data and said, if we look at the people who are lifelong, I suppose using statins or other kind of cardiovascular, cardiometabolic disease risk mitigation medications, what can we learn about the genetics of those people? I'd love if you could talk about that paper a little bit and what you learned, because I thought it was really interesting.
Sure. So I think that what we did was that in FinnGen study, which is a study of 500,000 Finns, or 520,000 Finns, which is 10% of Finnish population, which means that it's sort of a quite representative of the population here. So one of the things that we have for FinnGen, on top of having genetic screens on everybody, is pretty rich longitudinal health data. This means hospitalizations, this means all the cancers, this means actually all the cancer screening information, but it also means all the prescription drug purchases since 1995 are there. Wow. In the FinnGen database for these 500,000 people. So what we did in that particular paper is that we focused on cardiometabolic disease preventive or treating drugs. So we looked at statin usage, we looked at anti-hypertensive medication, as well as type 2 diabetes medications. And then we asked the question that if we simply map or run a GWAS on how many pills have I bought over my life for these, what does that tell about the underlying diseases? And we could show that we actually it's such a powerful phenotype that we could actually find quite a bit of new genes for these underlying diseases. So that was one of the outcomes of that. But perhaps that's not the most interesting one, except as an exercise showing that this medication data could be used meaningfully to study the disease. But the second thing was that we wanted really to try to develop tools to, when I start the new medication, if I had the genetic screen, could I predict my behavior as a user of the medication? And there are two different things that we were particularly interested in. Can we predict who stops the usage of these medications after a year or two, either because of lack of efficacy or because of adverse events? So that was interesting. And we could show that actually polygenic risk scores for the risk factor that we are treating with these medications, we see that if that risk factor is high for statin use, you're more likely to stay on the medication. So even though the individuals don't know their risk score for LDL levels, for example, they seem to be more often staying there. So that's good. So the second thing is that we looked at switching of the medication for some of these cases where it's clear that you switch from first-line medication to a stronger or more efficacious medication. And for example, for diabetes, it's clear that we can also use the type 2 diabetes risk score to predict who are the most likely ones to go from, say, metformin to stronger medications and eventually to insulin. So that was kind of interesting, because at the end of the day, it's kind of a combination of behavioral endpoint and there's underlying biology there. So it's a mixture of those two.
Sono Genetics will be at D-Farm in Philadelphia on September 16th and 17th, and we're planning on carving out space for creativity, connection, and a hands-on experience you won't find on the main agenda. If you'd like to meet with us to talk about your study, or if you're curious about what we've got planned, just drop us a note at events@sonogenetics.com. We'd love to connect while we're there.
Yeah, that's interesting. I want to ask about that almost like level 2 application of polygenic risk score. So you have early detection, and you described very well in both CVD applications as well as breast cancer. Maybe you could say more about what we know about how it could be used to predict prognosis or trajectory with or without medication, because I think that's a good example of if you can predict that somebody's going to need to go on to a second or third line therapy eventually, maybe you ask the question of is the outcome better to bring that forward for this group of people that has severe genetic risk? What do we know about that today, and what can we do to learn more?
So I think we can do a lot better than what we know today. So I think overall, at least for the past 20 years, the genetics has almost completely focused on modeling disease risk.
Yeah.
But what happens after the diagnosis, and what is the role of genetic variation or germline genetic variation after that, we know very little for almost all of the diseases. And this is partly due to the way that data sets have been, or biobanks have been put together. But I think now that we have more and more of these biobanks and biobank studies with decades of longitudinal data and with thousands of cases of many diseases, I think we are getting there to start answering the questions on different kinds of questions on the longitudinal scale and related to the diagnosis. What is how important are the genetic factors for disease progression overall? How important are they for stratified treatment or personalized treatment choices, adverse events of the treatment, complications, and I think more generally healthy aging or our trajectories over the life course? What is the role of genetic on different parts of our lives, given if we condition on the life course history that we have had? And I think that's going to be really exciting.
Yeah, it also has me thinking about some, maybe they seem like gnarly statistical challenges to me, but maybe you, as the card-carrying statistician, can enlighten me on how you think about this. But the presence of medications, it's really important to know about them, because if you don't, then it can really confound your downstream analysis, right? If the most severe group is also getting medications earlier, then they may end up looking like the less severe group because they're being treated effectively, right? How do you deal with that, with the potential confounding that comes from the introduction of the healthcare system doing its best to fix us?
I think that's a really, really important question, which we haven't really solved yet. So there's sort of that, like you said, most severe cases are treated, more strongly but then on top of that is that if you have a genetic liability to get the disease in the first place, you're more likely to get it earlier.
Yeah.
So when you look at what happens after that, you're in a different position. If I get a disease when I'm 30, I'm in a different position overall than if I get it at the age of 50, which is sort of the index event bias and what happens is that it may look like you said that the risk of getting the diseases may actually be in reverse associated with the risk of prognosis, and this really needs to be adjusted for. How to do that best depends on the disease and on the setting, really. But I think the key is to have as deep information about the treatment, about the disease severity is the key there.
Yeah, I mean, you really need that in-depth information that you get from integrated biobanks like FinnGen, right? Otherwise, I'm struggling to see a way that you can tease that apart. If you only know what happens at the end, then you're really blind to some of these confounders in the middle. Maybe you can talk about FinnGen. You've worked in FIMM and with FinnGen for a long time. For those who aren't familiar with it, it would be great to just give an overview of what it is, how it came about, the kinds of data that's collected, and in your mind, some of the most impactful things that have come out of it.
So, in short, FinnGen is a biobank research project. It's come together across Finland, so it has collected samples from all over Finland, 10 different smaller biobanks coming together to collect this collection. 520,000 individuals with genetic data in the background, as well as then link it to all the relevant health, more and more other type of registry data in Finland. And what I mean by registry data is that these are data sets, or these are data that's nationwide, covers the whole nation, and they have primarily been collected for administrative purposes. So that's why government has said that we need this data to understand what is the healthcare usage in different parts of the country and so on. So it's sort of been mandatory for any kind of healthcare provider to collect these. And some of these go way back. So the cancer registry, for example, was founded in 1953.
Wow.
All the hospitalizations go back to 1969. So that's 55 years of every single hospitalization in the country, with ICD coding for the diagnosis, operations codes for what has been done, and so forth. So it's sort of electronic database of all the health events, which is structured. That's important, and it's harmonized by definition. So that's in and we keep adding more of these type of data. The latest addition, which we really are just putting our teeth on right now, is that all the laboratory tests done in the whole country have been collected for the past about 10 years, and that's 211 million laboratory tests now linked to FinnGen data. So all kinds of lab tests.
Wow.
Now the question is, again, it's kind of related to your very good question about the prognosis or the statistical challenges related to prognosis, is that there's of course a reason for the healthcare system to take a laboratory test. And so we need to differentiate and try to model in why is this lab test taken. But I think we have 150,000 individuals with over 10 lipid measures over the past 10 years. So this shows the strength of the data in many ways. So this is public-private partnership. FinnGen is we have 14 different pharma partners that pre-competitively work together. So these farmers that are competing very much are working together in FinnGen with the academic world in Finland and all over. And that's one of the beauties of FinnGen, is really for us on the academic side to listen to what are the most important problems, challenges that the pharma is facing, thinking together how can FinnGen answer to some of these questions, or at least provide, help with the new medication development or the target prioritization, and or identifying sometimes even new targets. But so that's in short what FinnGen is about. And what has particularly interested me, which is a sort of a minority of all the analysis done in FinnGen, are the longitudinal aspects and sort of getting closer to the clinical translations.
When you think about that academic industry relationship, what are the big things that you see over the next 5 years, say, where when you get everybody around the table, everyone's looking at the problem the same way and saying, yes, we absolutely need to do this. Like I had Rory Collins on one of the early episodes of the podcast. This was back in 2020, and they were talking about proteomics at the time, and we've seen a huge wave across a number of biobanks. What is that today for you if you look at the next 5 years? What is the big, it could be technology or something else, but what are you thinking about?
For us, a challenge right now is that we see a lot of Finnish-specific associations, associations that we only see, either we only see in FinnGen, or at least the variants are so much enriched in Finland that it's difficult to study them elsewhere. So what we need, if we want to move on from simple associations, we need to understand at least something about the function relationship to the nearby gene, or if it's a coding variant, at least see that part. So what we are pushing on is various molecular profiling that we can do at scale.
Yeah,
and proteomics is definitely one of those things, but also cellular profiling and metabolite profiling, so that we have our own tool set to move ahead, at least the first steps in moving ahead in terms of defining the function of the variant and getting started on the path towards the disease. So that's a similar answer to what Rory Collins said, maybe with a little bit modernized tools now, and this is what UK Biobank is also doing. The second thing is that what we have in Finland, and this is really due to the legislation side of things. So early on, we had a Biobank Act that allows going back to the individuals. So I think the framework of having deeply profiled 500,000 individuals, that's a pretty good setting for downstream clinical trials, for example.
Yeah.
Focusing on certain combinations or certain profiles or certain risk levels or something. So that's another area that needs developing, but the sort of the basic framework is there. We've done a couple of pilots on this in FinnGen, and we are building on that. The third thing is really what we already discussed. This is the prognosis and moving ahead from simply looking at the risk, because from pharma perspective, we have preventive medications, but most of the medication is really focusing on treating people and understanding the processes of genetic and other background, and this treatment is an area that I see for the next years as an important.
What do you think about using polygenic risk scores in clinical trials? Is this something that we're going to start to see a lot more? The argument I hear for that is you can look at very rare or somewhat rare monogenic forms of disease, build a polygenic score that maps to that same pathway, for lack of a better term, and potentially increase the number of patients that could be treated. But you also hear the opposite argument, which is pharma companies never want to reduce the size of the addressable population if they can, and so running a polygenic risk score that identifies a really small fraction of the population is not as attractive as something like a GLP-1, for example, where you can treat absolutely everybody. I'm curious to get your perspective on how you think those factor into new drug development in the future.
In terms of the trials, we have a long tradition of at least on some level enriching individuals who are high risk for the disease, so that the follow-up for the trial doesn't need to be 10 years. So if it can be cut to 5 years, it's a huge change. And again, polygenic risk scores are definitely one tool in doing that, right? So I think we are going to see those. But I think more and more what we are going to see perhaps earlier on than looking at the phase 3 trials, or phase 2 trials, is really going into back of these biobanks, identifying really interesting genetic profile individuals and doing trials on those which are smaller scale, but again, polygenic risk scores are one tool there on top of that. So it's restricting variation, right? So, and focusing on the highest incidence group of individuals, and that's where it can help.
Yeah, that makes sense. As you look forward for the focus that your research is going to take, I'd love, as we close out here, if you could leave people with some of the major areas that you're going to be focusing on the next couple of years, so that they can hear more about how you're thinking about the world and follow your work.
I think one of the things is that we've been very good at piling as many cases as possible without so much focusing on the heterogeneity between the cases. I think we now with the very large biobanks, we can sort of go back and define our phenotypes more strictly, and that will help us understand some of the biology. That will help us also downstream in thinking of the sort of the personalized treatment options and so forth. So that's one thing. Related to that is the fact that in the world of polygenic risk scores, we've been focusing on trying to build risk scores that apply for everybody. But I think risk scores are good tools also to start understanding the concept specificity of the genetic risk. It's not, the genes are not operating similarly for everybody. And again, the large scale biobanks help us there. We can, do very traditional epidemiological questions. Does the genetic risk score work similarly in different age groups, sex groups, different backgrounds, ancestry backgrounds, or other maybe social variables? So this is an important theme that we need to understand better, and we have the tools now. The third, which is going to be perhaps the most exciting, is that we as a community need to really push these pilots towards clinical applications, and that will mean some large-scale trials as well to show that these tools work as they should. But really working with real patients, like we are now doing in the breast cancer study that we are running as part of the Intervene Consortium in Europe. I think that these are the type of things that we need more and more as a community if we want to continue to use that as an argument for funding our studies, that we will get to the healthcare eventually.
Yeah, that's right. With all of these in mind, when you look at things like Our Future Health, for example, in the UK, that is going genotype arrays first, is my understanding, but to a very huge number of people, 5 million is the target, which would be probably about 10% of the UK population. But if you did something like that in Finland, you could get nearly the whole population, right? How do you think about scaling up maybe to 10x the size that you are today versus going deeper and deeper into phenotyping and putting the resources there? Because if you had to choose, or if you were thinking about how to best pursue those, where would you put your resources if you thought about it for Finland specifically?
I would rather not choose. I would do both.
Yeah.
A subset with very deep phenotyping and profiling, and this is what we are doing in FinnGen as well. But at the time, I mean, we are getting close to the point where genotyping everybody would make sense. The question is really at which point of life and how to do that. And this is, I mean, our large-scale biobanks are getting old as well. UK Biobank or FinnGen people are not anymore young adults, and we need to be better at, particularly if we want to do predictive or preventive medicine. We need to profile those that are just learning what they are going to eat for the rest of their lives, or their exercise patterns, or so forth. So that's the time to profile them.
Great. Well, exciting vision, and thank you for rejecting the false dichotomy that you have to choose one or the other. It's perfectly fine to choose both.
Yeah, good. Thank you.
Yeah, thank you, and thanks everybody as always for listening. If you enjoyed this episode, then we really appreciate you sharing it with a friend. That's the most important thing. Thanks again, and we'll see you next time. That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Sanogenetics on Twitter, LinkedIn, or visit sanogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible. In particular, Amy Cousins and Sonia Shah, who produced the show, and James Pierce from Selected Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.